This is the Evolution Notes Part 5 on cladistics. We've already learned about speciation and how species may evolve over time to form a new species. Because of this, there are groups of species that come from a common ancestor. These groups are known as clades and can be represented by a diagram called a cladogram, as shown below. This entire structure right here, this tree, is called a cladogram. Clades can vary from small to large. This is a clade, and this is a clade, but the entire cladogram would also be a clade. Cladograms can contain organisms that are alive to present day, but they can also contain extinct organisms. It depends on what you're studying. Here are more examples of clades. Here's a clade. One branch is also considered a clade, but the bottom two are not considered clades. This one's not a clade because it's not including the common ancestor of these organisms, which is way down here. This one's not a clade because it's excluding one of the branches that came from this common ancestor, so therefore it's not considered a true clade. So these two are true clades, but these ones are not true clades. Essentially, cladograms are larger clades with smaller clades nested within them. Cladograms are diagrams that look like trees. They're sometimes known as phylogenetic trees that show both the similarities and the differences between organisms. They show the similarities by grouping them together in clades. They show the differences because the farther back in time you go, the more distant common ancestor they would share. They're more different from other organisms. If they belong to other clades from other organisms, they'd also be more different. The branching points are known as nodes. So here's a node, here's a node, here's a node, here's a node, essentially. And a node denotes a common ancestor that is shared between all organisms above that particular node. So if we look at let's say this node right here, this node is shared by, that's the common ancestor shared by humans, bonobos, and chimpanzees. Each node shows a speciation event where the common ancestor split into two or more species. On this cladogram, it's only showing two species splitting off, but there are cladograms that would show more than two. So it does not just have to be a splitting of two, it can be a splitting into three, into four. It's just more commonly two. The branching pattern of the cladogram is assumed to match phylogeny of that organism and therefore evolutionary origins of each species. So when we learned all that taxa, domain, kingdom, phylum, class, order, family, genus, species, that should correlate with what the cladogram is also showing. Today's cladograms are typically based off DNA and amino acid sequences. Those are homologies. The more they share, the more recent their common ancestor would be. The less they share, the more distant their common ancestor would be. We use computer software to be able to compare these sequences together. It's not typically just one sequence that's compared. It's typically more than one sequence that will be compared. That allows for you to find the most likely evolutionary divergence, the most likely cladograms. Cladograms are probable outcomes. They are hypotheses of what we think happened based on data. The principle of parsimony says that you'll choose the most simplest pathway that shows how evolution could have occurred. You will choose the pathway that requires the least amount of evolutionary changes that is supported by the evidence given. So we will look at examples on the next slide where parsimony has been applied. So here's hypothesis 1 and hypothesis 2. You can see that in hypothesis 1, the bony skeleton developed here, and then all organisms with bones developed after that. So sharks over here, which are cartilaginous, they don't have hard bones, would be considered the least related. And this particular cladogram, hypothesis 2, they have bony skeleton developing here, but then they have it developing here also way later in time. So that's a pretty big trait to develop, a bony skeleton. It would be highly unlikely that it would develop 2 times independently in evolutionary history. It has like essentially fish, some sort of common ancestor to fish diverging first, and then from that we have sharks and then bony fish. And so the principle of parsimony would say it makes more sense for the bony skeleton trait to develop just 1 time in history. So hypothesis 1 would be applied. You do need to be careful because you can draw cladograms in 2 different shapes. Here's shape 1 and here's shape 2, but they are showing the same information. The order of these nodes up here, which are called the terminal nodes, so the order of the organisms at the ends of the cladograms do not matter. They can be in any order. It's these nodes that show the common ancestor that matter the order. So in this cladogram, A and B share the most recent common ancestor, and C has the least in common. On this cladogram, A and B share the most recent common ancestor, and C has the least in common. So they are showing the same relationships. Each node is also rotatable, so you can like rotate these 2 organisms, for example, or C could be on this side. Same for this cladogram. It doesn't matter as long as it's still showing the same relationships. Here shows kind of the anatomy of these cladograms. Here's the terminal nodes. Those are the organisms on the ends. We have our nodes that show common ancestors. You do need to always have a root on your cladogram. And then whichever organism shares the least in common, so in this case gorillas, they are considered the outgroup. The outgroup defines the ancestral characteristics, and they have the most distant common ancestor. The DNA evidence used to build cladograms can be used to build many kinds of cladograms. This one shows humans and primates. This one has a time scale, which is kind of nice, but they don't always have a time scale to estimate when these divergences happened. If they don't have a time scale, it's still a relative time because the longer the line, the more time has passed. Here shows you, here's 2 questions. Please pause the video and answer these questions. Hopefully you came up with 8 for question 1. 1, 2, 3, 4, 5, 6, 7, 8 total nodes on this cladogram. And true or false, humans and chimpanzees descended from gorilla? That is false. They do share a common ancestor, but humans and chimpanzees did not descend from gorillas. In this particular slide, it's talking about how when we're looking at those changes in DNA sequences, we're looking at mutations. Mutations in DNA that have changed over time. The more mutations, the more time that has passed. If you assume that the mutations occur at a predictable rate, like throughout time, then you can look at a rate of time that has passed due to those mutations. They typically do choose reliable traits to be able to look at things like this. For example, mitochondrial DNA is found in a lot of organisms, all humans and primates for sure, and this DNA has been completely sequenced and it's known to be very reliable. Therefore, we can use this particular DNA evidence to create the cladogram. It's important for you to understand that even though we are using these rates as like a molecular clock to determine how long ago these events did happen, this is only an estimate of age because we're making the assumption that the mutation rate is predictable or constant, even though it's not. There's always variation in mutation rates, and that can vary based on a lot of factors. How long the generation is, the overall size of the organism, selection pressures acting on the organism, etc. Again, even if it does not show a time scale. It still is showing relative time. The longer the line, the more mutations have occurred because more time has passed. So the length of the line is still an estimate of time. Some cladograms are drawn to scale, but often they are not. Some sequences are going to be more conserved than others. Conserved sequences are sequences that have not changed very much over time. So when we look at a species, these particular sequences have remained identical or at least very similar over a period of time. Some species or some sequences are highly conserved, which means they have remained unchanged or very similar over extremely long periods of evolutionary time. There are hypotheses out there as to why some sequences are more conserved compared to others, because some sequences are known to be quite variable. One is that if it's in the coding region, it might have a really important function. For example, an important enzyme, or the genes that code for creating rRNA are very important. So these have changed very little over time. If it's in the non-coding region, they may just change very slowly, a very slow mutation rate. And so that would also lead to them being more conserved. Take a look at this cladogram and answer these. Oh, I didn't realize I was going to show you the answers. Okay, well, if we look at this cladogram, it says what animal is the most closely related to the squirrel. So if we find the squirrel here, that would be the guinea pig. For this question, what animals are more closely related to humans than to Old World monkeys? So if you find Old World monkeys and humans are down here, it's going to be everything above Old World monkeys. So gibbons, orangutans, gorillas, bonobos, and chimpanzees. Tarsiers share the most recent common ancestor with which animals? Let's find tarsiers here. Here's their most recent common ancestor right there. And they share it with all these animals above them on the cladogram. Elephant shrews, elephants, manatees, and dugong all share a common ancestor. Which animal diverged from the common ancestor first? So they're up here. And here's that common ancestor that they share. And the elephant shrew diverged first. Homologous structures, remember this is review, are structures that are similar because they share a common ancestor. It means that the DNA instructions that led to those traits are incredibly similar. Therefore, it's evidence towards divergent evolution or recent common ancestry. I always give the example of the forearm bones in birds. So we all have the same overall bone structure. A big bone, one big bone, two big bones, little bones, a little bit bigger bones, and then your final like phalanges or fingertip bones. And that structure is the same in a whale flipper, in a bird wing, in a lizard leg, in a cat leg, in a bird wing, in a bat wing, all the same. That's because they share a recent common ancestor. Analogous structures are the opposite. They show evidence of convergent evolution, where organisms appear to be becoming more similar over time. The DNA instructions that led to analogous traits are not similar. So here shows you some legs of different organisms that lack bones. They'd be analogous to these. Another example is like the wings of a bird or the wings of an insect. Birds have bones. Insects do not, so they do not share a recent common ancestor. The DNA instructions that led to wings are very different from each other. Those two winged events happened independently of each other over time. So analogous structures show evidence towards distant common ancestry. In the past, we looked at just morphological traits to figure out how organisms were grouped together within their different taxa, and therefore to build the cladograms. But over time, we learned that this wasn't always accurate. Once we were able to get DNA sequences and study comparisons of DNA or amino acid alignments, it allowed for us to no longer use morphology solely to create the cladograms. It allowed for us to use more specific, reliable evidence. DNA is always the best evidence, and so because of that, it caused us to have to regroup certain organisms. Some organisms were placed in the wrong clades and had to be moved to others. Some organisms needed to be merged together into similar clades. It really just depends on the ones that we're talking about, but we did have to reclassify many organisms in both the plant and animal kingdom. I'm going to go through an example. It's going to be the same example in your book. You don't necessarily have to know this specific example, but you do need to know an example of the reclassification of an organism. So I'm going to go through the figwort family. The figwort family is a group of flowering plants. They belong to the largest phyla of plants, which are called angiosperms. This family was huge until recently. They had a whole lot of genre in it, but these were all based on morphological traits, like shape of the flower, shape of the leaves, petal color, things like that. And the problem was that those similarities are superficial. Eventually, we realized that when we compared their DNA sequences of different chloroplast genes, that they were actually more different than we thought, and therefore it caused the reclassification of the figwort family. They had 5 clades combined into one, so these 5 clades had to be separated out because these were analogous traits, traits we thought originally were homologous because they were physical traits, but it turned out that they were actually analogous traits. Those traits that they had used had evolved independently in time. Therefore, it had to be reclassified into 5 different clades. Next, we'll look at an example where we build a cladogram. In order to build a cladogram, you have to have DNA or amino acid sequences. Typically, they're going to use computers for this, but we are going to practice one or a few this year by hand as well. So here we have 3 different organisms, A, B, C. At this point, we don't know which ones are more closely related. That's what we're trying to figure out so we can build a cladogram. You need to count the differences between A and B, between A and C, and between B and C in these sequences, count the mutations that have occurred, and then record it in the table in your note taker. So you should pause the video to do that. Once we record it in our table, which I just put in order at first, A, B, C, because I wasn't sure how many differences they were going to have, I then have to reorganize it based on who has more differences and who has least differences. You always want to try to organize the table so that the organisms with the least amount of differences, so the ones that are like more similar, are going to be the closest to the left, like in the top left corner. And then the ones that are more different should be towards the bottom right. So I have reorganized them A, C, B, and this side has to match A, C, B. Notice that there's a lot of X's. So they always get X'd out or not used if it's the same species. So A to A is the same species, so why would we compare them? They'd have no changes because they're the same. So that's going to be true of C to C and B to B. But also we would do that, the X's, for the ones we've already compared. So I already compared A to C here, so I don't need to compare it again here. Then we're going to use this information to build a cladogram. Please pause the video and try to build a cladogram off this information. Here's one example of a cladogram. Remember, yours could be different. You could have rotated these 2 nodes. You could have swung this out to the other side. You could have used the boxy ones. I'll try to draw it here with my pointer. So I would have like A here and C here and connect it like a box like that, like a U shape. And then coming out of the middle of that box, I'd have another branch coming down and over, and that's where I'd have B. And then my root at the bottom. But there's a lot of different ways to draw the cladogram, so remember, you might draw it differently than somebody else, but you could both be right as long as your nodes are showing the same information. And that's it for the notes.